of Cambridge in UK, where he studied the brain basis for delusion. And after that, he received a fellowship award which brought him to Yale and to further explore the maintenance of delusion. He joined the Yale faculty in year 2011, and he's currently an assistant professor in the Department of Psychiatry. And today he's going to share with us about his work on delusion and brain.
Yeah, so I guess the one thing I think is important to know about Phil is that, even though he's quite young, as you can see, he's actually extremely well known because he developed this model that you'll hear more about, quite early on in his career. I think if I'm not mistaken, maybe while you were a graduate student in Cambridge, that is this brilliant synthesis of, sort of the basic neuroscience model of, associative learning and sort of a pretty sort of credible pharmacological model of psychosis. And so he developed this very sort of complex model, and then over the subsequent years, he has steadily produced data, supporting it. And so this is a pretty sort of unique or quite unusual sort of story to see in psychiatric research. Both because of the integrative translational nature of the model and the consistency of his data to support it. So I'm really excited to hear about your new stuff, and welcome to the Martinos Center.
Thank you for having me. Thank you for the invite. So, as Daphne said, I'm interested in sort of translational or an integrative approach to psychiatry. And this is a slide that I actually borrowed from Danny Weinberger. And far be it from me to disagree with Danny, but he proposes that if we can just find the genes that predispose people to mental illness, we can understand the sort of cellular level dysfunctions that those genes are associated with, how those dysfunctions are manifest at the systems level in the brain, and somehow this sort of phenomena of psychiatric symptoms kind of come out. And I'd like to propose moving in the opposite direction, that if we can try and understand the symptoms of mental illnesses, we can maybe sort of reverse translate things back into sort of infrahuman species where we can make more invasive recordings. And I'd like to give you an example, one way that we've done this with delusions, delusional beliefs, sort of trying to model delusions in terms of how humans form causal beliefs, and they're driven by this process of prediction error, which you're going to hear a lot about today. So start with a delusion. This is our sort of DSM definition of a delusion, a fixed false belief based on incorrect inference about the external world that's firmly sustained despite what everyone else believes and despite contradictory evidence. There's lots of different reasons why this definition is wrong or unhelpful. There are lots of examples of how delusions aren't necessarily false. They're not necessarily based on external reality. The key thing for my talk and the model is that they're beliefs. And the idea is that if we can understand something about how healthy beliefs form, we might understand something about how unhealthy beliefs or delusions form. This is an example. It's one of the cardinal delusions. It's called the Capgras delusion. It's a delusion of misidentification. So this is a 74-year-old lady who turns up for psychiatric treatment because she believes her husband has been replaced by a different, unrelated man. And it drives her actions, right? So she refuses to sleep with the imposter in the same bedroom, she locks herself in a room, and she asks her son to procure a gun to protect herself. So just a bit of a structure of the talk. I'm going to show you how we've tracked causal belief formation using functional imaging, how we've sort of explored the effects of psychosis on belief formation and memory processing and changes in subjective experience associated with psychosis, and how we've used a sort of individual differences approach, individual differences in brain responses, behavior, and the severity of psychosis to form and to test a new model. And this has a sort of a long history, this idea of individual differences. This is a paper from an American psychologist in 1975 talking about how if one wants to implicate a particular psychological process in a phenomenon, then looking at individual variability in that process relating to the phenomenon might be a good place to start, a sort of a crucible to start constructing a new theory. And this idea has been developed at the neuroimaging level, looking at individual differences in brain responses to different tasks. To sort of test models that bridge psychology and biology. But paper, again in American Psychology, curiously, American psychologist by Stephen Kosslyn. So this is our model of how human causal beliefs form. The same processes that drive animals to associate rewards with stimuli in the environment might drive humans to form causal beliefs, associations between representations of causes and effects. we see the same pattern of responses when we arrange relationships between tones and rewards or punishers as when we arrange relationships between causes and effects that we present to our human subjects. So might they be explained by the same theories? And the sort of front-runner theory looks very overly mathematically complex. It was developed at Yale in the 1970s by Bob Rescorla and Alan Wagner. And it really is just an overly formal way of saying that we learn from our mistakes, or we learn most in situations when our expectancies are violated. That term in parentheses there is the mismatch between what we expect to happen and what we actually experience. And that drives us to learn new things, to update our beliefs. And if there's no prediction error, we don't learn anything at all. It's a nice little quote here from a review about prediction error in the brain by Wolfram Schultz and Tony Dickinson, saying that prediction error, that term in parentheses, relates to the very essence of learning. We don't learn anything when outcomes are perfectly predicted. And in a nutshell, our model of how delusions form, and I'll hopefully convince you why they're maintained, is that there's too much prediction error. Prediction error signals happen in patients' brains when they really ought not to. They correlate with meaningless events, and that leads patients to attend to and learn about those events, and that forms the kernel of their delusional belief. So in terms of the task that we're going to use, we're going to have an activation condition that engenders a bold response, and a baseline condition that has everything that the activation condition has minus one process, and of course that's our prediction error. And the magnitude of the difference between those two conditions is really that individual difference that we're going to focus on. And we're going to correlate that with the severity of symptoms that people experience in a psychotic state. So in the first study to try and localize where prediction errors might be happening in the brain, Paul Fletcher ran a study that was published in Nature Neuroscience in 2001, having subjects in the scanner learning relationships between fictitious drugs and fictitious side effects. So each trial they see a case study where a patient's taken a drug called Dugitil and they suffer Huxley syndrome. Now at the beginning of training, people don't have a particularly strong opinion about the relationship between these two things, so they're relatively surprised. But as you see more and more instances, that prediction error term gets smaller and smaller. However, we can show instances where Dugitil doesn't cause Huxley syndrome, and that's going to be associated with an increase in our prediction error signal. So that leaves us with three predictions about a brain region that might be coding or correlating with prediction error in some way. First, it should be most active at the beginning of training when you don't really have any particularly strong beliefs. Activity should decrease as you see more and more instances that confirm your predictions. And if we violate what you've learned, activity in that region should be re-evoked. And what Paul found was that a region of right dorsolateral prefrontal cortex satisfied all three of those predictions. And that was perhaps a little bit surprising because, as I mentioned, Wolfram Schultz recording from midbrain dopamine cells in monkeys showed that those cells actually show a very similar prediction error-like signal. That is, if rewards are unpredicted, those dopamine cells fire. If you show cues that predict those rewards, the dopamine neurons start to fire to the cues that are best predictors of the reward. And if you give less reward than was actually expected, you see a dip below baseline in firing. And I've highlighted those two different processes in two different colours because I think it's going to be important later on in my talk when we talk about delusion maintenance. Now we tried to replicate this effect and look at a more broad set of causal beliefs with a new task. We call it the food allergy learning task. We get subjects to come to the scanner, imagine that they're an allergist. They need to figure out the causes of allergic reactions in a fictitious patient. We show them meals that the patient's eaten, and then we show them whether or not he suffers an allergy. And their task is, when a particular meal of one or two or three different foods appears on the screen, they need to make a prediction about what they think is going to happen. Will he have an allergy or not? And they hold the button down for longer, the more confident they are they're making the right choice. So this gives us lots more cues that people can form beliefs about, and it gives us the opportunity to set up expectancies and to confirm or violate those expectancies. So this is one example of one of the studies that we ran. This looks at a phenomenon called super learning. So at the beginning of training, I show you that bananas cause the allergic reaction in our fictitious patient, Mr. X. Bananas paired with mushrooms don't cause the allergy, and that causes what we call preventative learning. The mushrooms must be overcoming the allergenic potential of the bananas. And then when I show you mushrooms paired with a novel food, the pair there at the bottom causing the allergy, that's super surprising. You get a double prediction error. And we can compare that to trials that are balanced for the presence or absence of the allergy and for the novelty and familiarity of the cues. And what we see is exactly the same region of right dorsolateral prefrontal cortex engaged in Paul's study, engaged in preventative learning, and engaged in super learning. So there's a nice relationship between our psychological process, prediction error, and activation in the context of this task in right dorsolateral prefrontal cortex. And perhaps it's not that surprising because that region is projected to pretty widely, directly by those vitiate dopamine cells that Wolfram records from, and also indirectly through the basal ganglia. the bold response, of course, is quite sluggish, so what we might be measuring is sort of synaptic inputs into that region. So not necessarily spiking in that region itself. We've also engaged in a meta-analysis where we looked at least 73 papers, that number's gone up now, on prediction error, published in the literature looking at punishment, reward, and also more sort of non-valent tasks like ours. About half of those papers show accumbens activation or caudate activation, putamen activation, but critically, quite a few of them show activations in DLPFC. So again, perhaps not quite so surprising. Well, we wanted to use this relationship to try and tell us something new about causal learning and causal belief formation. So we picked on a phenomenon from causal learning that's sort of slightly problematic for standard error-correcting theories. It's kind of controversial. It's called retrospective revaluation. It's a situation where you change your belief based on your belief about cues based on trials when those cues weren't actually present. There's an example from Sherlock Holmes here. Is there anything that you'd point my attention to the curious incident of the dog in the nighttime? The dog didn't do anything in the nighttime, and that was the curious incident. So updating your beliefs in light of something that didn't happen. Prediction errors can't really apply, according to the standard model, to absent cues. So in our task, we engender it by showing you that hamburger and cheese together cause the allergy. Hamburger alone doesn't cause the allergy, and that makes you change your opinion, update your opinion about the absent but expected cue, the cheese. So it must have been the cheese that was causing the allergy in this case. And there are sort of at least two different competing solutions and cognitive models for how this takes place. There's a probabilistic model that just says people function as intuitive statisticians. They add up all the instances when allergies happen and cues happen. And crucially, prediction error isn't particularly important for this model. It doesn't figure in the probabilistic model. We can also modify our sort of standard Rescorla-Wagner model and say that when absent but expected foods are not presented, there's a representation retrieved, and prediction error acts on that retrieved representation. So prediction errors can drive learning about absent but expected cues. Both these types of model predict the same behavior, so can we use neuroscience and our imaging marker to adjudicate between the two? Basically, right frontal activation at the time of retrospective revaluation would favor our sort of more associative prediction error-driven model, and if we didn't see right DLPFC activation, that would favor a sort of non-prediction error-driven or perhaps probabilistic model. So this is the standard design, like I outlined. Hamburger plus cheese cause the allergy. Hamburger alone makes you change your belief about the absent but expected food. And because it's imaging, we added a novel stage where we either confirmed or violated that inference that had taken place. So it would be surprising to see the cheese not causing the allergy, and it would be unsurprising to see the cheese causing the allergy. So across 3 phases of training, we're looking for right dorsolateral prefrontal cortex activation at stage 2 to implicate prediction error in this revaluative process, and then we're going to look at the magnitude across subjects of that activation in the context of having one's expectancy or learning violated. And this is what we found. So much like in our prior studies, right dorsolateral prefrontal cortex activation occurred at stage 2 during that revaluation process. When we violated that learning at stage 3, we saw activation in the same region, and there's some nice overlap between the two. And those two things correlate with one another. So both frontal and striatal activation during revaluation correlates with the amount of surprise that people experienced at stage 3 when we violated their expectancies. So there's a nice model for engendering expectations and looking at prediction error. Well, I was working in a psychiatry department, so I had to kind of make it relevant to mental illness in some way. So if associative accounts explain causal learning, can we use them to explain these erroneous causal conclusions that patients draw, these delusional beliefs? What does our prediction error brain marker look like in psychotic states? And can we use fMRI and this causal learning paradigm to tell us something new about psychosis? Very, very early on in psychotic illness, as I mentioned, patients' attention is drawn to irrelevant things in the world. Background stimuli seem much louder or brighter. They form links between things that are merely coincident in their mind, and they might form their delusions to account for these sort of bizarre experiences. So aberrant prediction error signals might provide a mechanism for these sorts of things to take place. This is by no means a new idea. I think I traced it back at least to John Locke, the British associationist philosopher, who talked about the aberrant association of ideas in our minds driving us mad. You might see some resonance with Geoffrey Gray's work and David Hemsley's, and also more recently Satish Kapoor's work. What we're offering here is a slightly more mechanistic model for how aberrant salience might come about. Crucially, these things happen a lot before patients turn up for psychiatric treatment. So they're a difficult to identify population because they often don't think that there's anything wrong. In fact, many patients quite like the very early phases of psychosis. They feel they've got a sort of insight into the world that other people maybe don't have. So they're very hard to study. And we thought that ketamine might provide a useful experimental model for this phase of the illness, because we can control, in healthy volunteers, we can transiently induce these similar symptoms using this drug. We can do it reversibly and safely, and we can do it in the fMRI scanner. So ketamine is an NMDA receptor antagonist, acts on the main sort of excitatory transmitter in the brain, glutamate. It was developed in the 1960s as an anesthetic, but its use in human adults has been curtailed massively because of what we call emergence phenomena. That is, when people came out of the anesthesia, they experienced beliefs and hallucinations that were very, very redolent of those psychotic symptoms that we've outlined. This is an example. This is a graduate student from Yale, given ketamine whilst I was interviewing them. They said every time I left the room, they thought another person dressed in my clothes was coming back into the room, doing my job. The person was a bit older in age and weighed a little bit more. Now, I've definitely aged and I've definitely gained weight since I moved to America, but not on that sort of scale. And if you think back to the initial example that I gave, this is very, very similar to our old lady who thinks that her husband's been replaced by an imposter. Obviously, I wasn't married to this person, but there's some sort of surface level familiarity changes. It seems like a different person's coming in and out of the room. Other things that ketamine does, subjects feel much like our patients, that their hearing or their visions change, things become much more salient, things in the background becoming much more important. They become much more aware of external noises, people talking, phones ringing outside of the room, and they think that the telephone call is about them. And the object of their gaze changing, surrounding things seeming fuzzy, and things that they focus on seeming hypersalient. There are also cognitive effects. They have lots of a sort of flight of ideas, of things being connected with one another. And these sort of simple ideas or delusions of reference, feeling like things are personally significant. So feeling like people are looking at them or talking about them behind their back. And the people at the scanner, the radiographers who run the scans for us, maybe being spies. And that they sometimes even lose insight. And we studied these phenomena using a relatively novel design for fMRI. So one of the things that ketamine does is induce nausea and vomiting. And vomiting in the scanner is a really bad thing, both for patient safety and for sort of maintaining collaborations with the radiology department. So what we did was give people a low subpsychotic dose of ketamine whilst they were in the scanner performing our causal learning task. We then brought them out of the scanner and turned the dose up to one that really induced those clinical phenomena that we're interested in. And the key thing that we can do is do the same thing on a placebo visit, counterbalanced in order, and we can kind of compare the neural responses on ketamine and placebo and use those neural responses to predict the severity of the ketamine-induced symptoms that people experience. And these were subjects' brain responses on the placebo and ketamine visits in our key region of right dorsolateral prefrontal cortex. So when we violate people's expectancies on placebo compared to unsurprising events, there's this nice engagement of that region. However, on ketamine, surprising events, there seems to be less of a response, and unsurprising, run-of-the-mill, highly predictable control events seem to have more of a response. So there's a sort of a blurring of the distinction, if you like, between responses to surprising and unsurprising events. Some evidence, perhaps, that our prediction error process is inappropriately engaged while people are on this low dose of ketamine. Linking it with psychosis, what we found was that people who were most surprised by expectancy violation on placebo had a worse time on ketamine. So they had more perceptual and attentional disruption, and they also had more of those sort of simple ideas of reference, thinking that people were spying on them. In terms of the actual ketamine effect predicting subsequent ketamine responses, the more that right dorsolateral prefrontal cortical region was engaged to inappropriate events, the more severe the perceptual aberrations and delusion-like ideation people experienced. So the more of that blurring of the distinction between responses to surprising and unsurprising events, the more severe the psychosis-like symptoms that people experienced. This is our ketamine effect. Ketamine's a model of psychosis. How good of a model is it? So we replicated the study in a population of first episode psychotic patients. These are people very, very early on in their illness. They're obviously having some problems and have been referred to our first episode service, but not sort of chronically ill, haven't been ill long enough in order to meet a diagnosis of schizophrenia even. And this is how their brains responded to the task. So compared to control subjects, patients with first episode psychosis show a response in that right dorsolateral prefrontal cortical region that's very, very similar to the effect of ketamine in healthy volunteers. And just like our ketamine effect, the more that right DLPFC region was engaged, the more severe the delusion-like ideation was that people experienced, both in terms of the raw response and in terms of a sort of simple computational model that we built that used subjects' confidence in their predictions as a way of estimating the amount of prediction error. Now, This is where I moved to Yale, and we sort of tried to develop the model to look at the tenacity or fixity of delusions. There's a sort of strange thing that patients can't be talked out of their beliefs, even in the face of overwhelming contradictory evidence. And sometimes confronting patients with contradictory evidence like that can be worse for their prognosis. Their delusions have a property of what we call elasticity. That is, if I tell you that it's not at all possible that the Queen of England is actually living in your house or following you around, what they'll do is, instead of taking that evidence on board, they'll encompass the evidence and sort of involve me, implicate me in the plot. And people who do that remain deluded for longer, they respond worse to CBT, and they just have a worse prognosis. So how can a prediction error model, that's essentially a very plastic learning mechanism, explain the fixity of delusions as well as their formation? So I'd just like to sort of walk you through an example. This is an example from Peter Chadwick, who was a psychologist in the UK who actually suffered from psychosis, and he's written really elegantly about his delusional beliefs. And he talks about how he sits on the top deck of the bus, and he goes to the right-hand side, looks out of the window and notices that lots of the shops on the right-hand side of the road are painted green, and it strikes him as a definite pattern. He moves over to the window seat on the left and notices things on the left-hand side are painted red. And he decides that things must have been arranged that way by this nefarious organization who are giving him sort of clues as to what might be going on, and he needs to work out the meaning. So this is the kind of thing we're trying to explain. And the way we're going to explain it is using a phenomenon called memory reconsolidation. I don't know if you're familiar with it. It's something that's experienced something of a renaissance in recent years, at least in behavioral neuroscience. This is a phenomenon whereby we can condition animals that a tone predicts an electric shock. We can put animals back into that conditioning setting and remind them with the tone of the shock. That renders the memory of the shock-tone association into a labile state, so it's susceptible to being disrupted by extra behavioral training, different drugs, and electroconvulsive shock. Why would you want to do that? Well, it suggests that memories are more sort of dynamic than simply providing a way of storing information. They can be updated in light of new information, and they can be involved in sort of prospection and planning for the future. So reconsolidation might be a way of sort of strengthening or streamlining or honing in on the key features of a memory that are important to be maintained. And there's this process of sort of automatization or habitual sort of memory formation that reconsolidation might underpin, forming schemas from original episodic events to sort of more semanticized representations of what's happening. And I like this idea that reconsolidation might drive the sort of transition from knowledge about a specific event to a belief about those kinds of events in general. Just to show you some animal data, the drug manipulations aren't particularly important. What I want you to focus on is that dotted line. This shows that after 2 sessions of conditioning, merely reminding an animal of the conditioning context can strengthen their memory for that context, can strengthen their responding. So simply engaging with a memory might lead to that memory to being more strengthened. In terms of our kind of prediction error cartoon, sort of thinking about those initial Wolfram-Schultz data that I showed you, there's this peak in responding to cues that are predictive of rewards. There's a dip below baseline in firing that engenders a sort of extinction learning process when what we've predicted doesn't actually come true. And what happens perhaps when lots of cues are presented is that reminder process is excessively engaged, such that that extinction learning can't possibly compete or override the rumination that you've engaged in. So there are sort of 2 processes, as I outlined on Wolfram's data. There's this red process of being reminded and thinking about and making predictions, and then there's this blue extinction process that engages a sort of new learning that actually the relationship between the cue and the outcome no longer pertains. And what I'd like to suggest is, in the context of an aberrant prediction error system, you get lots and lots of reminding about the original reinforced situation, such that even when the organization don't end up getting you, the extinction learning can't possibly compete. And what gets expressed in behavior is the thing that you've thought about, reconsolidated, and ruminated upon the most. So if we think about our example from Peter Chadwick, you might think that there was a sort of peak in prediction error firing at the time that he noticed the red doors and the green doors. There's a sort of insight relief that he has when he figures out that the organization must have been explaining things or arranging things in this particular way, such that every single time he subsequently experiences odd prediction error feelings, the organization is re-evoked and used to explain those things away. And it's so powerful that even when the organization don't get him, even when the sort of consequences of his prediction don't come true, that extinction learning process can't possibly compete. And this was sort of laid out previously by Carl Jaspers, who's the sort of father of modern psychopathology in psychiatry, talking about how when delusions are formed, the trail is blazed and preparedness for significant experience permeates almost all contexts. The dominant delusion motivates the apperceptive schema for things that happen to you in the future. So what I'm going to show you now is an experiment that we've done, it was recently published just last week actually, where we tried to test this idea using our ketamine model. It's kind of a complex study, but I'll walk you through it. Hopefully, if you have any questions, I'd be happy to kind of clarify. So it's a two-stage study. Initially, we have subjects come to the scanner, we capture their prediction error brain responses using a phenomenon called blocking, which I'll walk you through. And then in the second stage, they come back, they have a behavioral study, a placebo-controlled behavioral study of memory reconsolidation of fear memories. So in the first stage, what we're going to do is show them that a food causes the allergic reaction. That food paired with a novel food, bananas and the mushrooms, cause an allergic reaction of the same magnitude. And that should, if they've learned that bananas cause the allergy, that should block any new learning about the mushrooms, such that if I show you mushrooms causing the allergic reaction, that should engage our prediction error response. So there should be not very much learning, not much prediction error here, and lots here. Exactly the same layout of trials as we've used previously. Then a month later, we invite people to come back to the lab and undergo this ketamine study. The first day, we give them fear conditioning, where a prepared stimulus, a picture of a spider, predicts the delivery of an unpleasant loud noise. Another prepared picture predicts no loud noise. Then we give them ketamine and we remind them on a single trial of the cue that previously predicted the loud noise. And we track their responses to that cue the next day in extinction. So simply reminding someone in a psychotic state of a memory that they've previously formed about a salient event ought to, if ketamine is inducing inappropriate reconsolidation, it ought to strengthen the memory the next day. And if prediction error is driving that process, then individual differences in prediction error brain response ought to predict that strengthening effect. This is what we found in terms of our brain region in our right dorsolateral prefrontal cortical prediction error response. During the blocking trials, there's a predicted attenuation. No learning takes place, maybe a little bit of learning. And then when we violate the blocking, when we show those mushrooms causing the allergic reaction, activity in that right dorsolateral prefrontal region is re-engaged. Behaviorally, subjects don't form a strong causal belief about the mushrooms, the blocked cue, compared to the control trials. And there's a nice correlation between the amount of prediction, the amount that they learn about the blocked stimulus, and their brain responses both during the blocking trials and when we violate that blocking. So there's a sort of nice internal consistency, again, between the brain response and their behavioral responses to our task. In terms of the subsequent ketamine study, they learn that one of the cues predicts the loud noise and the other cue doesn't predict the loud noise. What I'm showing you here is ketamine and placebo conditioning on the day before we give them either ketamine or placebo, measuring their skin conductance responses, so the amount of sweating that they show in response to these different cues. So they've learned an equal amount about the two different cues in those two different settings. We then reactivate the cues and bring people back on day 3 and look at responses to those cues in extinction. And what you see is the cue that was re-engaged with and reactivated while people were in a psychotic state on ketamine shows an enhanced response that's slower to extinguish relative to the placebo cue. If we just randomly play them the loud noise, you get stronger reinstatement of responding to that ketamine reactivated cue. So some evidence that experiencing a memory whilst on ketamine in a psychotic state induces strengthening of that memory. We also assess the severity of subjects' symptoms. So we're looking at using the clinician-administered dissociative state scale to measure those aberrant salience experiences, just like we did previously. And the amount of strengthening that we see correlates with the severity of the symptoms that people experienced on ketamine. The strengthening was also manifest at the level of their sort of conscious subjective ratings of the cues. So they find the cue that was reactivated under ketamine much more arousing than the cue that was reactivated under placebo. And like I said, they show this sort of more implicit skin conductance response enhancement to the ketamine reactivated cue. And interestingly, their conscious subjective ratings of how arousing the cues were correlated with inappropriate engagement of the striatum in our causal learning task, and the more sort of implicit GSR measures correlated with engagement of that canonical right dorsolateral prefrontal cortical response. So showing this response inappropriately to trials that really ought not to be surprising correlates with that strengthening effect that we saw. The data that I just showed you pertain to this final violation stage. I'm also interested in those individual differences in what's going on during blocking, so whilst you're sort of seeing this irrelevant stimulus being presented. And again, what we see is a correlation between inappropriate engagement of our prediction error response during the blocking trials that correlates with increases in the strength of the memory that was reactivated under ketamine relative to the memory that was reactivated under placebo. Now you might say that the ketamine experience and being rendered psychotic at Yale by an experimenter for a short period of time at least is aversive, and so you're going to get generalization from an aversive memory to an aversive experience. So we tried to replicate the effect in a separate group of subjects with an appetitive learning paradigm. So what we're doing here is just showing subjects neutral cues, just these sort of random stimuli that predict delivery of juices into their mouth using a pump, a computer-controlled infusion pump. So one cue predicts orange juice, another cue predicts blackcurrant juice, and a neutral cue predicts just the delivery of some water. What we're going to do is reactivate one of the appetitive cues while subjects are on ketamine, and we're going to bring them back the next day and track their responses in extinction to those cues again. And we're going to make two different measures. We're going to get them to anticipate how much they're going to like the cue, the juice that's followed by the cue, so make an explicit rating of what they think is going to happen next. And we also measure the amount of sucking pressure that they apply across the end of the tube that's in their mouth. So do they suck in anticipation of a tasty juice? And the idea is that ketamine reactivated cue, they should predict that they're going to like the juice more if ketamine is strengthening their memory for that cue juice association. And they're also going to apply more sucking pressure in response to the cue when it's shown on the screen. And this is exactly what we saw. So both in terms of the explicit measures of liking, the cue that was reactivated under ketamine is liked more, and they show more of that anticipatory sucking response than cues that were reactivated under placebo. And just to show you that it's strengthening rather than simply a failure of that single extinction trial, showing you the responses to those cues at the end of day 1 and then the beginning of day 3. So in between these two sort of columns here, there's going to have been our reactivation procedure on either ketamine or placebo, and I think you'll agree that they're showing an increased response to the cue that was reactivated under ketamine rather than a lack of extinction learning taking place. So it's really something to do with that initial reminder process rather than the extinction process. So just to summarize, I've shown you a model of delusion formation as aberrant causal learning. It's driven by prediction error. It's processed by PFC in particular, although of course the midbrain and striatum are also engaged. I've implicated PFC in particular in ketamine-induced and endogenous psychosis. I've shown you that this sort of paradoxical or puzzling strengthening of memories under ketamine, and preliminary data suggests that that's associated with inappropriate engagement of our prediction error signal. Where are we at in terms of our translational model? So with Jane Taylor, I've kind of reverse translated some of this work into rodents. So what we've shown is ketamine impairs blocking on in mice and in rats. So ketamine induces inappropriate responding to the irrelevant or neutral blocked cue. And ketamine also strengthens reactivated memories in rats. So we do exactly the same procedure where we give the rats ketamine right before we reactivate their memory for a context, and we see strengthening the following day. So some evidence at least that the behavioral phenomena that I'm talking about that seem to be really clinically relevant translate back across species. And with that, I'll thank my collaborators, both in Cambridge and now at Yale, and thank you for your attention.